The roadmap of our DNA. You may not know much about the Human Genome Project because it hasn't made much noise in the media. In the science world, however, the topic is shrouded in controversy. To some, this project is doubted as a fruitless endeavor, but to many scientists, this is the holy grail of biological advancement. But what is it? Well, the Human Genome Project is the sequencing of our DNA to learn and study genetic disease. This is the most important project of our generation so far. In a publication in 1993 from the Science Journal, scientists defined the project as an international effort to develop genetic and physical maps and determine the DNA sequence of the human genome and the genomes of model organisms. The ideas that I will discuss involve the timeline of the project and the issues facing the scientists, the science behind the Human Genome Project, and the application of the results in real medicine. The Human Genome Project got off to a rough start due to timeline projections, budgets, and ethical dilemmas. In a publication in 2003 from the Science Journal, evidence of the beginning shows that the first serious discussion of the possibility of sequencing the human genome was convened in 1985 by Robert Sinsheimer. The idea began with a rebellion against the view that biological research must always be hypothesis-driven. Due to the conflict caused by this, the project didn't actually start until 1990. The projected cost of this project was $3 billion. A large problem was acquiring funds to support the project. In a publication of Social Studies of Science from 1996, the dilemma of acquiring funds to map out the human genome was compared to the statement that said, "Grants are not awarded to count the grains of sand on the beach." Research was done in small amounts, and once that gained considerable credibility, an overwhelming amount of grants were awarded to fund the project. To start the project, data needed to be collected from the human population. In a publication from 2001 in the journal Social Studies of Science, the diversity of the Human Genome Project was discussed with a specific goal in mind: to collect and preserve the genomes of isolated indigenous populations. The consequence was that researchers were worried that it would be unethical to just take DNA samples from these isolated populations. However, this viewpoint was overthrown by the idea that it would be unethical to not preserve a certain group's genome, which is in fact their genetic history. Now that we've covered the origins of the project, we can move on to the science. The Human Genome Project has been a means to map out our DNA and study its base pairs and code for those proteins. The Human Genome Project, when boiled down to a science, is merely observation of our DNA. According to Daniel Kevles and Leroy Hood, authors of the Code of Codes, Scientific and Social Issues in the Human Genome Project, DNA must be broken into ordered smaller fragments. This is a process called physical mapping. Our DNA is divided into approximately 3 billion base pairs that each have about 20 different variations. These pairs create a chain-like fence that makes up our DNA. The problem concerning this is the time it takes to analyze each base pair, given that there are 3 billion of them. In an article published February 10, 2011 on DNA sequencing technology, Elaine Marty stated that in 2005, a variety of new engines for DNA sequencing that were radically different from the capillary sequencers used to sequence the human and model organism genomes became available from several different manufacturers. The process of coding for proteins involves looking for certain proteins that are linked to certain genetic diseases. In a Nature publication from 2011, scientists suggested a total of 30 to 40,000 protein-coding genes. These protein-coding genes represent genetic variations in our DNA that can make someone more prone to developing a genetic disease such as cancer or Alzheimer's disease. Now that we've gone over the science, I will explain the application in real medicine. The results of the Human Genome Project can be directly applied to real medicine as an effective form of preventative treatment of genetic disease. It can also be applied to the cross-analysis between different species, and ultimately be used for evolutionary innovation. The big picture of the Human Genome Project will allow scientists to look out for certain diseases and remove their possibility from our DNA. In a Nature Genetics article from 1999, scientists claim that massively parallel measurement strategies promise to revolutionize how we study and ultimately understand the complex biochemical circuitry responsible for controlling normal development, physiologic homeostasis, and disease processes. We can better understand how genetic diseases work and attack them before they come to be by changing bits of our DNA. Scientists can cross-analyze genomes between animals and humans to better understand diseases that occur in both. Cross-analyzing would potentially allow us to save endangered species in the same way we would save ourselves. The Human Genome Project will allow us to permanently change our DNA. Whether or not you think that's playing God is a completely different problem. This means that our DNA can be altered by our own hand, giving us control over our own evolution. The human genome has been successfully mapped out at this point, which is a great achievement to add to the records of human history. The project has lasted for over 20 years, in the face of many obstacles. The science has proven to be valid, and the application of the results are more than promising. To reiterate, the Human Genome Project wasn't just some experiment. It's our ticket to longer and healthier lives. It is the culmination of hard work from many scientists all over the globe. We have found the holy grail of biological studies, and it would be highly unethical for the sake of the human race to not take advantage of this opportunity.